Hello, today I am going to be taking a brief lecture on G-stimulatory pathway. Before going to that, I would like to talk about the hormone receptors. There are three types of hormone receptors. Cell membrane receptor is the first one. In that we have protein hormones like FSH, LH, etc. And we have catecholamines. These hormones cannot cross the lipid bilayer, hence they have to act on the cell membrane receptor and give the intracellular signaling. The second one is cytoplasmic receptors. Example is steroid hormones. These hormones can cross the lipid bilayer and can act on the receptors on the cytoplasm. And the third one is nuclear receptors, and the example is thyroid hormones. They can cross the lipid bilayer and act on the nuclear receptor. Now let me give an outline about the receptors on the cell membrane. There are three types of receptors on the cell membrane. First one is 7-pars receptor. That is the G-coupled hormone receptor. G-coupled hormone receptor. And the second one is 1-pars receptor. That is the enzyme linked hormone receptor. And then we have the ion linked hormone receptor. What is 7-pars receptor? 7-pars, this is a cell membrane. The receptor is like this. 1, 2, 3, 4, 5, 6, 7. This is 7-pars receptor, and 1-pars means this is the 1-pars receptor. Today, as I mentioned, I'll be talking only about G-coupled hormone receptor, and in that I'll be talking only about the GS stimulatory. We have GS, GQ and G inhibitory. Today I'll be dealing only with GS pathway. So G protein is made up of a beta gamma subunit and an alpha subunit. This makes a trimeric protein. So when the beta gamma subunit is together with alpha GDP, alpha subunit, this is the inactivated form. So when this is getting activated, as I mentioned, epinephrine is one among the catecholamines which acts on the cell membrane. So let this be a beta 1 receptor, and let this be a cardiac tissue. So what happens is that when epinephrine is acting or stimulating the G coupled receptor, this gives an intracellular signal. Because of this signal, this alpha GDP now gets activated and gets converted to alpha GTP. So I repeat, when the hormone receptor complex is made, GDP is displaced with GTP. This detached alpha subunit now activates the adenylyl cyclase, which is a second messenger. So what is the second messenger? Second messengers are intracellular signaling molecules. These are the intracellular signaling molecules released by the cell in response to a exposure to an extracellular signaling molecule. These are the first messengers, like hormones are the first messengers. Here we have the epinephrine as the first messenger. And now this adenylyl cyclase converts ATP to cyclic AMP. Activated cyclic AMP now activates the protein kinase A, which is the main component which causes the phosphorylation of various proteins. For example, we have taken a cardiac tissue here, and epinephrine is the hormone. So what happens that when there is an increase in cyclic AMP and increase in protein kinase A, this protein kinase A goes and activates the L-type calcium channel and causes the calcium influx. There is a calcium influx. This calcium goes and stimulates the sarcoplasmic reticulum to release more calcium. So what is happening here? There is a calcium induced calcium release. Thus there is the contraction. We know one of the functions of epinephrine on the cardiac tissue is contractility. Just make sure that in the cardiac tissue, when cyclic AMP is increased, we have contraction. But in vascular smooth muscles, when cyclic AMP is increased, we have relaxation. This is a very small topic, but this is a very, very important topic. That's all about G stimulatory pathway. Hope this makes sense. Thank you.